In this video, we will discuss about the mechanism of STAT3 activation. So, in the previous video, we have understood that in the absence of RAF1 and EGFR, it is there is a activations of this STAT3, and that prevents the cell death or keep the cancer cells resistance against the cell death. So, they wanted to find out how these activations of STAT3 happens. So, they start with the canonical regulator for this STAT3, that is IL6ra and this Jak1 Jak2. So they knock down this IL6ra, Jak1, Jak2 and check whether it will has any impact on the cell proliferations and death. So when this RAF1 and EGFR are present, at that time we do not see any significant change in the colony formations, and also when these are been knocked down, that is RAF and EGFR are been knocked down, at that time also they do not see any difference in the colony formations when this ILR6 and Jak1, Jak2s are knocked down. Whereas in the control where STAT3 is knocked down, they are not able to see any colonies. So they do not find out the role of these regulators on the STAT3 activations. And they also checked it for the STAT5 knockdown. In that case also they do not see any changes. Thereafter they check for, they explored the SRC family kinase, and there they are able to find out there is a high phosphorylations of this SRC419, and this they find out using the immunoprecipitation assay. So then they check the effect of this SRC phosphorylations in the presence and absence of RAF1 and EGFR. So they are able to see that in the absence of RAF and EGFR, there is a high upregulations of this phosphorylated PSRC, and they checked it for the two independent cases, and they found the similar results, indicating that this SRC family kinase could be the reason for the activations of STAT3. Then they check the gene expressions of the SRC family kinase, and they see that these high phosphorylations of the SRCs occurs because of the upregulations of the enzyme, because of the upregulation of the gene FYN. So before that they check this phosphorylated Y419 using the immunoprecipitant also, and they are able to see that in the case where RAF1 and EGFR is knocked down, there is a high upregulations or higher upregulations of this phosphorylated SRC at 419 positions compared to the case where RAF1 and EGFR R1 are present. So this confirms their finding from the immunoprecipitant that there is upregulations of this phosphorylated SRC419. Then they check the gene expressions data of this SRC family kinase, and they see that there is a high expression of gene FYN in the case of this RAF and EGFR1 knockdown. So now what they did is they silence this FYN gene using the using this silencing RNA against FYN, and they can see that there is a very low productions of this FYN in that case, even when this RAF and EGFR1 are knocked down. And so, and also this phosphorylated STAT3 is also low in that conditions. Now since the phosphorylated of the STAT3 is low, means the activations of the STAT3 will be low compared to the control case. Then they check the colony formations in these silencing FIN conditions, and they are able to see that in the case where FIN is silenced and also the RF and EGFR are knocked down, they are able to see less colony formations. And also even if the RF1 and EGFR1 are not knocked down, they are able to see less colony formation or almost negligible colony formations, indicating that this STAT3 is the one that helps in the resistance against the cell death of this pancreatic cancer. So, and here it seems like that FIN levels are almost similar, whether in the presence or absence of RAF and EGFR1. And however, from the gene expression data, they checked that high phosphorylations of this Y149 could be because of the high expression of the genes FIN, but it is not showing in the Western blot somehow, or they have not quantified this difference in the expression levels. But when this FIN is knocked down, they are clearly able to see the reduced level of or reduced resistance against the cell death. So I hope you understood how they check the mechanism of the STAT3 activations, and they find out that this SRC family kinase plays a role towards this STAT3 activations, and it's the STAT3 that provides the resistance, activated STAT3 that provides the resistance against the cell death, even whether RAF1 and EGFR are present or they are not present. That is seen from this panel E. So I hope you understood these results that shows that helps them to find out the mechanism of STAT3 activations. If you need to understand more about these experiments in detail, you can go through the reference that has been provided in the description. So that's all for this video. Thank you for your time.